Hello, Dean of Mars. Hello, Destiny Faith. So Prunia, hi. I'm still waiting for a couple of people to come in. Hello, Jimmy Souza. Has any one of you been here before? Do you know how many people I should expect? Hello, Jane Beth. So yeah, um, I don't know. How long should I be waiting? I don't actually. Maybe just one more minute. Okay, this seems to be it. So, at least the first wave of people have come in. Thanks for being here. Hi, Fab, RNI just joined. Oh, now they're coming. What's that? Okay, great. Okay, so I will just start. My name is Dennis. I'm German. I'm a neuroscientist, um, and I have um, been doing neuroscience for about 13 years now, which includes my undergrad thesis. And I did my undergraduate study in Cologne, which is in Germany, and it was on alternating courtship calls in fire-bellied frogs, which was a fun project, to be honest. Um, My so I did a degree. I got a degree that is not in existence anymore. It was before the European Union made the bachelor and master system available across Europe. So I did what's called a diploma in Germany, and I did that degree. I got that degree in biology, and my main subject was animal physiology, and I also was examined on biochemistry and developmental biology. And I also did a genetics course that, if I had completed it, meaning if I went to the tests, I could have been I could have become a major in genetics too. But then I decided not to. Um, from my undergraduate, hey, hello, Cherry Tomato, Sketch Art Pencil, Ivan, what's that, Biru Boy? I don't even know how to pronounce most of these things, but I'm glad you're here. Um, so I just explained that I did my undergrad in Cologne, uh, and it was already on behavior and neuroscience. And then I went on to do a PhD in Bielefeld, which is also in Germany, with my PhD advisor Hans-Joachim Bischoff and a co-advisor Martin Egelhoff, and that was on optic flow in zebrafinches. And you heard that right, it's zebrafinch, not zebrafish. That seems to confuse a lot of people, but it's a bird, it's not a fish. Optic flow is basically the image that you see on your retina when you move through the world. Um, So it's like the motion, the overall motion of the images on your retina while you're going through that. And yeah, zebrafinches use optic flow for depth perception. So the way images of objects move across the retina at a given speed and direction tells you how far away they are, gives you some sort of depth sense. Hello everybody who just joined. The Addictive photographer. Oh, interesting. Yeah, so that was my graduate thesis. Hello. And then I moved on to do a postdoc in the United States. How many years I spent in school? More than I could have. So the diploma degree back in the day, hello, it's Mrs. Science, okay. So the diploma degree was supposed to be about 6 years, and I think I took 7. But that's obviously different from today, where you do a bachelor's degree and a master's degree before you join a PhD, or at least that's how it is in Europe. I'm aware that in the United States you can start a PhD with a bachelor's degree. So it depends on where you, in which country you're in, and what system you're studying under. So yeah, from my PhD I went on to do a postdoc in Cold Spring Harbor Laboratory in the United States. It's on Long Island, if you don't know it. It's become infamous by the racist remarks and misogynist remarks of Watson, who has been the director of that place for a long time. That's Watson from Watson and Crick. And what I did there was working again on behavior and underlying neuroscience, and the topic was olfactory learning. So we know that olfactory learning plays a role in mate choice in mice, so that was quite interesting. And we also knew from the literature that noradrenaline plays a role. Noradrenaline is a so-called neuromodulator, so it's released from a central piece or from central area in the brain, and it goes into almost all other brain areas, and then it does things like increasing the ability of the neurons to change. So it increases neuroplasticity, as we say, and that allows the animals to learn. And we looked at that in a physiological study, imaging calcium signals in the so-called olfactory bulbs, which is the first stage where the information from the nose comes into the brain. So that was very interesting. Then I moved back to Europe. I'm now in Portugal, and I did 3 years of postdoc here, working on, still again on mice, this time running in mice and what the cerebellum, how the cerebellum coordinates limb movements in this context. I didn't get far with that study because I was working mostly on methods. The lab has its own tracking system that uses a video or machine vision to track the limbs of a mouse on a treadmill. And I've been working on that part, and I've also worked on a new setup that was supposed to, or it did in the end. So it was integrating the lab's system of recording mouse movements on a treadmill and video recording that together with optogenetic stimulation, which means the mouse is genetically altered in a way, either by viruses or by breeding, to have neurons in the area that you're interested in respond to light. So if you put a little glass fiber into the brain in the right position, then you can make these neurons fire, or you can inhibit these neurons. So that's optogenetic, and you can do that in the awake running around mouse. And on top of that, I was doing electrophysiology, which means you put in like little electrodes into the brain, and then you can record the electrical activity of the neurons, also while they're running. Hello, breathe a trip. So yeah, that was my project as a postdoc in Portugal. And last year in August I quit, and since then I've been mostly doing whatever I want. I'm taking a year off, or well, it was supposed to be a year. It might not end up being a year. Living off my savings. I'm in Lisbon, in the capital of Portugal. Right, so I'm responding to Breather Trip's question, where in Portugal I am. So yeah, I'm in the capital, close to the river. Yeah. So where was I? Yeah, I finished my postdoc, and since then I've mostly been working. Oh, hey there. It's okay, just chill. So from ending, Jesus, I got confused. So yeah, I started doing science advocacy. I'm chill. It might not seem like that, but I'm chill. I started doing science advocacy with the March for Science, which was in 2017. And the thing was that, so we were a small team getting together, trying to make this work in Portugal. And yeah, that was a fun experience, and I think there was a lot of science advocacy energy built up, at least in me. So after the march, actually, so the team disbanded, and so I wanted to continue with science advocacy, but I was the only one from the team who wanted to do that. So I started my own project called Science for Progress, and I've been working on that since. And since I left my postdoc, I've been doing that most of the time. I have a podcast for that, and we are running a rotating curation account on Twitter, where every week you have a different person talking about things related to scientific progressivism in a broader sense. And finally, I am trying to build up a freelance career. I really like to do scientific editing. I like editing manuscripts or grant applications, these kind of things. So I'm available for that. I'd like to do neuroscience consulting, and I really need to find clients right now. So that's that. And finally, in that context, I'm making videos for YouTube, and I also publish them on Facebook about scholarship. I just finished the first one, and it was published on Thursday. So this was a rundown through my life so far. Do you have specific questions on any of these things already? I saw one comment on my profile on Instagram in the Instagram post. Um, from No Punk in the Gym. I didn't see him join or her, but it was a fun thing. They're calling me a legend, so thanks for that. Um, loves my science literacy interest. Yeah, I hope everybody's listening to my podcast to hear what that's about. And so this person is majoring in neuroscience. Can it easy to make a career in science cartoon illustration? Oh wow, that's a difficult question. I don't really know. Um, Science, I actually, no, I just can't tell you. I don't know. I know one person who is doing designs, science designs, but that's more like schematics and stuff that are supposed to go into papers. I know that some of his designs made it on the title of big scientific journals. Um, but I mean, I can't answer for that, so sorry about that. How hard is it to find a science job here in Portugal? I think it's hard. There is not much funding, but so the good news is that in Portugal funding is rising for science. Um, I had it kind of easy because when I applied for a postdoc position here, I knew that there was an opening, and I knew some of the professors here at the Champollion Foundation. Um, so it was kind of easy for me to find something because I knew people. I'm not so sure how easy it is elsewhere. I think it's hard anyways. But I mean, it also depends on what you mean with a job. Do you mean a postdoc? Do you mean a PhD? Or in industry? I don't really know. I only know that, so when you're talking about industry, I know there is a pharmaceutical company called Bial who came to our lab and gave a presentation on what it is like to present or to apply for industry jobs. But other than that, I'm not really well versed in that, which is something I should change because I'm going to apply for jobs now. PhD probably. Okay, still in the last year of high school. Okay, so then you have time. So for you, things may change. I hear that the Portuguese government has pretty ambitious plans to increase science in Portugal. If they do what I hear they want to do, it's significant. So by the time you get out of undergrad, you might be this might be a very different situation. Okay. Do you want to hear more about science, or more about science advocacy, or more about scholarship? So, as I said in the profile, I'm here to talk about all three of these things, but I haven't prepared much, so I'm open to any suggestions. Yes, so all three of them. Or was that on science? Science, okay. So my science focused on behavior and physiology in in all of the cases, so every topic. And early on it was for me more about the animals, and what the animals can do and how the physiology supports the behavior of the animal. It was not so much about the translation to, for example, medicine or something. I do think that that's a good cause, and if the work that I do would have translated to medical purposes, that's great, but I'm more of a fundamental biology guy. And the second thing that interested me is methods and how you can apply them to reach your goals in science research. So one of the earliest, my first real science research thesis was with my undergrad in undergrad, and it was on frogs and how they do their mating calls. So if you have a lot of male frogs around a pond, then they're all shouting because they want to attract a female. And the problem with that is that if everybody is called at the same time, then the females have problems locating the one they're interested in. So they have this system where when they know they would be calling at the same time as another frog, they would inhibit the next call and then just after a short break start calling again. So that's a quite interesting phenomenon. And what we did was a behavioral experiment where you have the frog in a pond, or not in a pond, in an aquarium, and we gave it a testosterone injection so that it would be interested in calling. And then we added a loudspeaker and a synthesizer, and I programmed the synthesizer so that it would it would play calls of frogs, basically. So the good thing about these frog calls is, we were working on the oriental fire-bellied frog, which you might find maybe in China. And the good thing about those is that they have this very simple call that is basically just a tone repeated several times with a fixed rate. So it would go like, and there is literature out there where they actually calculated how both the temperature of the water and the size of the frog influence the frequency of the call and the rate at which the frog would call. And then I implemented that into a little program that would make the synthesizer create these naturalistic sounds. So I made this little program and I played it to the frogs, and I was able to replicate what they found in the wild, which is what I just explained. So you can make the frog start calling just by turning on the synthesizer, and then the synthesizer is calling and the frog is calling, and because they're not calling at exactly the same rate, you would have overlapping calls at some point, and when that moment is reached, then the actual frog would inhibit the next call and then start calling a little bit later. So just to avoid that they would be calling at the same time. And this project was supposed to then, they are easy to be manipulated. Yeah, it's quite funny. I hope it has to do with my stimulation being very naturalistic. So the next thing is a little bit of a Frankenstein experiment. Um, Basically, we removed the brain and then we had it in a jar, basically. And we were able to record the electrical activity in cranial nerves, which are large nerves that go in and out of the brain and then go into the face region, for example. And there is one that goes where the signal for breathing goes through, and one where the signal for the glottis goes through. And then there's a third one where the auditory signal comes in. So we stimulated on the auditory nerve and recorded from the breathing and the calling nerve. And I was able to record fictive calling, as it's called, which means there is no real calling because it's an isolated brain, but you can still see the signals for calling, and it was kind of creepy. Unfortunately, after I managed to do this, it was time to hand in my thesis, and this particular advisor didn't have the funding to keep me around, so that's where that ended. Which is sad. I actually think that especially the behavioral part was worth a publication, but it never came to that. Do you have questions about the frogs? Yeah, creepy isolated brain, exactly. But it's kind of it's amazing that you can take It's amazing that you can take out a brain and put it in a jar and it still stays alive for quite some time, actually, just by keeping it cool and having it in a fluid that has enough oxygen. You could not do that with a mammalian brain or with a bird brain. You can only do that with these amphibians, for example, who are cold-blooded and are more resilient to the tissue becoming cold. Because if you keep them warm, they will die quicker. Okay, so that was that. Then the next project was my graduate project, which also had pretty amazing methods. And I also think that the topic was quite interesting. So basically the problem is that you want to see, so when you're a bird and you're flying around really quickly, and Australians may know, you're welcome, I'm glad you heard something new. So the next thing is my graduate study, and the problem was that if you're a little bird and if you're from Australia, you know that zebra finches are not only numerous, but they also like to fly into the crowns of trees. And clearly what they want to do is avoid flying into the tree or flying again or colliding with other birds. Oh, hello, eLife Sciences, interesting. Hi, Aaron. So the problem that they have is that they cannot use stereo vision for depth perception. So the reason for that is that other than, for example, in the human head or in the head of other animals with forward-facing eyes that use depth perception a lot, the eyes are too close together, and they're also directed to the sides, which means that the part where the vision overlaps is very small, and it only works in a very close distance. So for pigeons, there is actually behavioral data on this, and they only can use their stereo vision for about 15 centimeters. and the zebra finches are much smaller than pigeons, which means that their stereo vision probably only reaches a couple of centimeters. So how do they see in depth? Well, there are other animals that face the same problem, which are even smaller, and that are flying insects. And our collaborators were working on blowflies at that time on that kind of topic. And the idea is that these animals use optic flow. So the motion of the image on the retina while you're moving through an environment to estimate depth to objects and to get an idea of the 3D composition of the environment. So what they do is move in very specific ways. So basically what you can imagine optic flow, how it works, is that you don't compare images of the two eyes, but you compare images of two time points that are where the image is taken from slightly different positions in space, and that works best if you're moving in a straight line. So that's when you get most of the depth information in your optic flow. If you make a movement with the head like this, then there is basically no depth information in the movement. Bifocal. You mean stereo vision? Is that? I'm not sure what you mean by bifocal. If you move in a straight line, then you get depth perception, and you can see that in insects, in flying insects, that they tend to move in straight lines rather than fly in like smoother curves. And that's exactly because, and the idea is that that's because they want to get depth information from this movement. So we were interested in whether the birds also do have a similar strategy in gaze. And what we did was build a flight arena which had a wall that they had to fly around. So if they wanted to get from one point in the arena to the opposite side, they would have to fly around that wall, which means they would be forced to fly in a curve. And as we said, a curve is not good for depth perception in that moment. Now birds are much larger than blowflies, even the zebra finches, which just have 25 grams. So they cannot turn their whole body as fast in mid-flight as insects can. So instead, the body does like a curved. That's I don't know, butterflies. Crazy. They don't fly that fast, I think. At least not in a close distance thing. They might fly fast when they travel long distance, and then they do that at a height where all this is not a problem. Why is my camera is dimming? I don't know why this is. Okay, sorry. So we had the birds fly in this smooth curve, and we wanted to know whether they still do something to their gaze. No, we didn't do hopping. So we didn't do. So we were looking at the orientation of the head while they were flying in that curve, and what we found was that the bird would hold the head in a constant angle compared to the world. So not compared to the body, but to the world, which means that the head wasn't doing the same smooth curve flight, basically, that the body would do, but it would try to avoid the optic flow from this rotation movement and to clean it up so that the eyes would mostly see the optic flow from the translation, so the straight movement. So we think that that's a hint that the birds also use optic flow for depth perception. And that again was followed up with an electrophysiology study where the animals were anesthetized, and we then put them into an LED monitor, which was pretty big, and that was yeah, so that's a different topic. I will come to that later. Do birds have a built-in compass? So what we had was like a spherical, almost spherical monitor, and we could project. So it had LEDs around, and they we could play videos on this monitor. So you can basically, it's basically, how do you say it? It's more or less like a virtual reality thing, where you have a surround monitor, and then we played a reconstruction of the movies from the real flight in the bird. So again, we had the trajectories of the birds flying through the arena, and we had the head movements. And then we could take this movement information and place it in a computer model of the arena. And from this we created these videos that were displayed on these more or less spherical, no, not holographic, monitors. So while the animal was anesthetized and inside of this monitor, we could do electrophysiology and record the response of the neurons to these flights, which was pretty hard to do, I must say. And we got some interesting clues out of that, but because it was the first study in that direction, it was more of an opening of a possible field. And not like a finding which would be the end result of a longer study or something. So that was my graduate thesis. You were asking about magnetoreception in the birds. And indeed, while I was a graduate student in the same lab, there was a postdoc who was working on magnetoreception in the zebra finches. And he found that they are able to do it, but they cannot tell all four directions, so north, east, southwest. They could only know whether it's on a south, in the north axis or on the east-west axis, if I remember that correctly. So they do have a magnetic compass, that's correct. But that doesn't help you to navigate in three-dimensional space. It only allows you a navigation on a long distance. So it gives you a sense of direction, but it doesn't allow you to fly around an obstacle in your environment. You need vision for that. So that was my PhD study. Are you still interested in more neuroscience, or do you want to hear something about Um, the other things that I do, I see hearts coming up. Is that for the science? So more science? Okay. Yes, sonar would work too. Echolocation, like in bats. That's true. I'm actually cited quite often by people who work on sonar. So that particular, my behavioral study is cited often by people who work on sonar in bats. Okay, so the next science story of my life was my work at Cold Spring Harbor Laboratory in the United States, where I was working on mice. And there, the idea is that mice learn the identity of other mice through their smell, and that's how they remember them. So for us, maybe the face is super important. Yeah, I don't know about migration in birds. I'm sorry. I just saw something about how they are moving earlier now because things are getting warmer too early. But I don't know more about that. So yeah, so the mice. The thing is that we wanted to know where in the brain olfaction and olfactory memory begins and how it happens. So there were studies from the early 1990s, maybe late 1980s, in sheep, where they found that if you have a ewe, so that's the female sheep, that would not accept a lamp, that you could make it accept the lamp by stimulating the cervix of the ewe, and then presenting the lamp. So that's kind of a weird thing, that you have to stimulate the cervix for that. But they already know that this recognition in lambs too, lambs and sheep too, is based on olfactory learning. So they looked at the olfactory bulb in sheep, and what they found was that first it's necessary that there is a noradrenaline surge into the main olfactory bulb. So the noradrenaline is a neuromodulator that is in the center, that's released by an area in the center of the brain into many regions in the brain that induces things like plasticity or increase the signal-to-noise ratio, if that means something to you, of the neural signal. Basically what that means is that signals are easier to differentiate for the animal if there's noradrenaline around. So we were interested in the neuroplasticity part of it, and the question is now, where in the main olfactory bulb do we have these changes? The way the signal is transmitted through the main olfactory bulb is you have the receptor neurons in the nose, and they would send the signal through their axons into the main olfactory bulb, and then onto so-called mitral cells. You don't need to remember the name, but these cells would then send the signal further to the cortex. And when my then advisor was a postdoc, he found that noradrenaline release during presentation of an odor would change the response of these neurons to that odor. So that's basically learning. Yeah, I don't know. My nose doesn't work very well, to be honest. I've had too many I have too many allergies, I think. And what he showed was, so his thing was to do electrophysiology in the main olfactory bulb from these mitral cells, and he would present an odor for a couple of times just to see a baseline, to have the baseline response of these neurons, and then he would couple the odor stimulation with an electrical stimulation of this noradrenaline releasing area. And then he would do another couple of recordings without the stimulation of noradrenaline. And that's how he could then compare the response to the odor before the treatment to the response to that same odor after the treatment. And then he saw that they responded less to that odor, which is an interesting result. And he was able to show in a behavioral experiment that male mice that received the smell of female urine during this treatment would later choose the urine that was not presented with noradrenaline. So they put them into a so-called wine maze, which is basically a hallway that then splits into two, and then they can choose which hallway they want to go, left or right. And in one of these sites, there would be the smell of the urine that was presented during the experiment, and on the other side there would be the smell of a urine from a different female, and they would significantly more often choose to investigate the new female, which is an indicator that they remembered the smell of the urine from during the experiment. And when I was a postdoc in his lab, the idea was to look. This is basically in the mitral cells, which are the second step. So we wanted to know whether you could find this change in response even earlier. So we looked at calcium signals. So this is now imaging, not electrophysiology. And what you do there is you have a genetically altered mouse that expresses calcium indicators in the olfactory sensory neurons. And what a calcium indicator is, it's basically a molecule that binds to calcium, and when it does that, it changes its form and becomes fluorescent. So if there is calcium around and it changes its form, and then you shine blue light on it, then it will shine green light back. And that it's very dim signal, but you can detect it with a good microscope. And that's what I did. The good thing about doing this in these so-called olfactory sensory neurons, which are the first neurons to pick up the chemical signal from the smell, is that they all come together and send their axons in very large, relatively speaking, neural structures, so that you can see the signal so you can choose a very large field to look at and still recognize the different odor responses. And then you would have like a pattern on the surface of the olfactory bulb that indicates the response to the odor, which is, so I'd love to show images actually to make this more clear. Anyways, so what I did was basically the same experiment that I explained before, where my advisor used electrophysiology, but now I don't have them. But but now with calcium imaging. So I presented an odor for a couple of times, and then I would do the stimulation of the noradrenaline releasing system during odor stimulation, and then I would do another couple of imaging rounds to see whether there is a change between the activity before the treatment and after the treatment. And I actually found the same thing, which was quite astonishing, because, I mean, these are the neurons who detect the chemical signal in the air. I would have actually expected that nothing would happen at that stage, but this was quite astonishing. So what we think now is that there is something happening in these large structures where the axons all come together to then put the signal onto the mitral cells that go then signal to the cortex, that would make so there's something happening there. There are more neurons involved in that cluster, so it could be that they actually change the signal in the axon before it reaches the mitral cell. So that's quite fascinating, because usually you think of memories as, oh, hello, pretty smart science. Usually you think of memories as these complicated things, but you can also think of memories as a very simple change in the physiology at just one single synapse. And that's, hey. So yeah, so that was quite a quite cool story. I like that in the end. It was pretty hard to do. For a couple of reasons. It was technically difficult. So yeah. I would talk more. I have yet another science story, actually several, but the hour is almost over. So do you have more information? So sorry for everybody who just came in just now. I think in my podcast or somewhere I said the wrong time. I said it would be 4 p.m. Lisbon time, but it's 3 p.m. So I hope I didn't. So I hope you're not here because of my. You're not here late because of my problem. Can you quantify smell? Well, in principle you can, if you use a gas spectrometer, but I've never done that myself. But that's basically what you often see on TV, for example, where they have a gas sample and they run it through this machine, and then you have like a plot where there's a peak at different points, and then depending on, I think, the molecular weight of the molecules in the smell, you get the peak at a different position, and the size of the peak gives you an idea of how much there is in there. I guess that's how they, for example, also register the amount of carbon dioxide in the atmosphere. If you're talking about quantifying an odor stimulus to have it precise, you can try to have the mechanism to be very how you say it, clean. Do we smell same thing that other animals do? Do we see the same colors that other animals do? The answer is no, we do not. We identify the same things, and we do share different receptor molecules. But for example, if you look at the human genome, you have, I think, about 300 receptor molecules, whereas mice, I think, have 1,000 roundabout. And then it depends on how many of these receptors you have in the nose and these kind of things. So in general, if you ask about perception of things, it's a pretty close guess, a pretty good guess to say no, they do not perceive things the same way. They will be able to differentiate the same things to some degree. Although, again, if you have more receptors, you will be able to differentiate more different odors than if you have fewer. It's just it's like having more colors in your eyes, then you can differentiate more. Mice seems to be more foodie versus human. Is that a is that a ratatouille thing? That's actually what they're talking about. That's why the rat is the good cook in ratatouille. Okay. Any more questions? We're almost done. I'll do like a minute or more, something more. Is there a quantification for the same? I don't understand what is the same in this question. Yeah, so what will they smell? So if they have more receptors, then it's probably more intense. It's hard to say really. So the comparison is difficult between mice and humans, because humans might have fewer receptors, but they have way more cells in the olfactory bulbs. The olfactory bulbs are a small part in the human brain, but they're still bigger than a mouse olfactory bulb. What exact work are you doing? Yeah, you missed. Is there a quantification for how closely they can differentiate between two very close smells? You can test that, and it's different for every odor. But it's a typical test that you also do in psychology with humans, where you ask the animal to identify which odor it is. And you do that by rewarding it if it pokes the nose in the right place for each smell. And then you get a very interesting S-shaped curve, sigmoidal curve. So if you have a mixture of both odors, might be, for example, 50-50, then it's really hard for the mouse to differentiate. And the more so, but if you have 100% one and 100% the other, then it becomes easier. But you do have to train the animals to do that. Usually, two very similar smells, the difference is not that relevant in everyday life for the animals. What exactly am I doing? Yeah, so just to recapitulate, I'm a neuroscientist. I've been doing research for 13 years, starting with my undergrad thesis, and I've done a PhD and 2 postdocs, and I am now, we have 20 seconds remaining, and right now I'm a freelancer. I quit the postdoc last year in August. So if you want to know more about me, follow me around on Twitter, go to my website. Okay, now I really have to say goodbye. 10 seconds. Okay, thanks for joining, and yeah, have a nice day. Bye-bye.